Hello my dear students. So today we are discussing a very important topic, that is the urine formation. The urine formation, where this process takes place in our body. You know, you have a special organ for that. This is the kidney. But kidney have a special type of cells. You already know that this is the nephron. Nephron is a structural and functional unit of the kidney. But what is the main function of the kidney? The main function of the kidney are the urine formation. You know, the urine is a very important substance for the human beings for our survival, because through the urine we are excrete the harmful nitrogenous substance, mainly the urea. But how the urine formation takes place in your kidney? I explain this process into the 3 steps. Number one, this is the glomerular filtration. Number one, it is the glomerular filtration. Number two, the reabsorption. And number three, the secretion. All these 3 steps are happening inside the nephron. But how it is happening? Now one by one we are discuss this topic. So now you are go to the first topic, that is the glomerular filtration. The glomerular filtration, you know, this process are happening inside the kidney. We already know that in our blood lots of nitrogenous substances are present. It is required to eliminate from our body. But how this nitrogenous waste material produced in our body? You know, all the cells in our body have some chemical reaction always takes place in there. This is called the metabolism. Due to metabolism, some nitrogenous substances are produced like urea. This urea is the main excretory product of the human beings, right? But when the blood are mixed up with the urea or some other substance, they ultimately go inside the kidney. Inside the kidney, we already know that the nephron is there. So now the blood takes place a particular position, that is the glomerular part. This one is the glomerular part. This one is the structure of the nephron. This is the first part of the nephron, that is the glomerulus, and that corresponding the Bowman capsule. In here, you know, the 1100 to 1200 of blood is purified in every minute. So the nitrogenous waste containing blood, they are enter into our this glomerulus part by the afferent arterioles. Afferent arterioles is a special type of blood vessels, right, which now enter into the glomerulus. They have the all nitrogenous waste, but a special structural modification is there and that particular site. When the afferent arterioles enter into the glomerulus, their diameter are decreased. But why their diameter are decreased? Because if the diameter of the decrease of any pipe, that time the pressure of the fluid are increased in that particular site. Same thing are happening in that particular portion. Now the blood are go inside the glomerulus. In here, the blood now pass the 3 layers to reach the Bowman capsule site. But what is these 3 layers? Number one, that is the endothelium of the glomerulus blood vessels. First of all, you can see this picture. In here, this is the endothelium layer of the glomerular blood vessels. Second layer, they are cross the epithelium layer of the Bowman capsule. That means this is the green color portion. It is the epithelium layer of the Bowman capsule. But between the 2 layers, the basement membrane are present. This one is the basement membrane. Now the blood which have the nitrogenous waste, they can pass into this 3 layer. After that, they reach into this Bowman capsule site. Bowman capsule site. So after the ultrafiltration and passing these 3 layers, they enter into the Bowman capsule. But you know, the epithelial cells of the Bowman capsule, they have a specialized cell. They have the specific name also. This is called the podocytes. In this picture, this is the podocytes. These podocyte cells in the Bowman capsule arranged in that manner that some spores is there in that particular site, and these fine pores have the very important role for passing that filtrate from the glomerular blood vessels to the Bowman capsule. These minute pores are called the slit pores. These minute pores are called the slit pores. In that particular position, the ultrafiltration takes place and glomerular filtrate are passed on to the Bowman capsule of the nephron. But what is the rate and what volume of filtrate are produced in that particular time? This is called the GFR. That is called the glomerular filtration rate, or the GFR, What is the value of the GFR? This is bear in mind, very important question. 120 5 ml blood, 120 ml of filtrate are produced in per minute. If you have calculated these things in per day, so it is a 180 liter. So it is the 180 liter per day. This is the GFR rate. So this is the first steps of the urine formation, that is the glomerular filtration. Now you are go to the second step. In that particular position. Now this process are called the reabsorption. But what is the mean by the reabsorption? Reabsorption means when the ultrafiltration takes place, so huge pressure are built up, so some important substance which is passed on in the Bowman capsule, but this is very important. But why we are release this substance from our body? So this important some substance again reabsorb in our blood. This is called the reabsorption. You know, I give a common example, that is the GFR rate is a 180 liter per day, but urine are formed, that is the 1.5 liter per day. Where the 180 and where the 1.5 liter, so huge gap is there. So this 99% filtrate are absorbed. 99% filtrate are absorbed in there. But in where? This is the main site, that is the PCT, or the proximal convoluted tubules. You know, in the PCT, the two type of absorption process takes place. One is the active reabsorption process, another one is the passive reabsorption process. In the active reabsorption process, the glucose, Na plus and amino acid are reabsorbed in the blood. On the other hand, in the passive reabsorption process, you know, the nitrogenous waste, some amount of nitrogenous waste and the water molecules are passively reabsorbed from that particular site into the blood. So this is called the selective reabsorption. But why? Because in selective sub-molecules only can reabsorb, but all the molecules not are reabsorbed, right? So now we are go to the third step of the urine formation. This is the secretion. A first question arises in your mind, where the secretion takes place. The main site of the secretion is the DCT. You know already what is the full form of the DCT? That is the distal convoluted tubules. you know the H plus, K plus and some ammonia that is present in the blood, right? But these substances are now enter into the filtrate, but not via the glomerulus. They directly pour up into the filtrate in the DCT portion. So this is called the secretion process. The secretion process are very important. You always bear in mind the what is difference between the absorption and the secretion. In the absorption process, the material are transferred from filtrate to blood. On the other hand, in the secretion process, the materials are transported from blood to filtrate. And this, the secretion process have the most important aspect in our body, that is that the ionic and acid base balance in your body fluids, that is takes place by the secretion process, right? So this is the three important process we are discussed in that particular topic, that is the urine formation methods, and we already understand that in the urine formation takes place into the three important steps, that is the number 1 glomerular filtration, second one reabsorption, and number 3 that is the secretion process, right? So if you understand this topic, so please you like this button, and if you are new in this channel, please you subscribe this channel for upcoming next video that is fast coming to your sites. Okay, thank you very much.